Okay, real talk. How many times have you told yourself you were going to do a 48-hour fast and made it to about hour 14 before you were standing in front of the fridge at midnight negotiating with yourself? Yeah, me too. Here's the thing nobody in the fasting community wants to admit. Most people can't sustain the kind of extended fasting that actually moves the needle on cellular repair. Not because they lack willpower, because they're human. And the brutal irony? The science has quietly moved on. While the influencers are still selling you on the romance of suffering through a 3-day water fast, researchers have been uncovering something far more interesting, something that doesn't require you to feel miserable. There is a specific ratio of amino acids, the building blocks of the protein you eat every day, that can flip the same biological switch that fasting flips. Same outcome, completely different path. No starvation, no energy crashes. No brain fog so thick you can't remember your own name. Just your body doing what it was always designed to do, cleaning itself from the inside out. And today, I'm going to walk you through exactly how it works. Stick with me, because by the end of this, the way you look at your dinner plate will never be the same. Let me ask you something. When you think about fasting, what do you think is actually doing the work? Most people would say, not eating, giving my digestive system a break, burning fat for fuel. And those things are real, but they're surface level. The actual mechanism, the one that's responsible for the cellular anti-aging effects that have scientists genuinely excited, has almost nothing to do with calories. It has everything to do with signals. Tiny chemical messages your cells are constantly reading, interpreting, and responding to. And one of the most powerful responses those signals can trigger is something called autophagy. I know, it sounds like a complicated word, but the concept is almost poetic once you understand it. Auto, self, phage, to eat. Your body eats itself, but not in the scary way that sounds. Picture your cells as a busy workshop. Over time, that workshop accumulates junk, broken machinery, defective parts, worn-out equipment that's just taking up space and slowing everything down. Autophagy is the cleanup crew. It comes in, identifies everything that's damaged or past its prime, and recycles it, breaks it down for parts, either repurposes it or clears it out entirely. And when that cleanup crew is doing its job properly, you think more clearly, you age more slowly, your immune system works better, your mitochondria, those little power generators inside every cell, become more efficient. The cleanup crew is essential. A Japanese scientist named Yoshinori Osumi spent decades figuring out exactly how this process works at the molecular level. In 2016, he won the Nobel Prize for it. So this isn't fringe science. This isn't a biohacking blog post. This is Nobel Prize-level, peer-reviewed, been studied for decades biology. Now here's the piece that changes everything. For a long time, scientists believed autophagy required one specific trigger: total caloric deprivation, no food, full stop. That belief made sense at the time. Every time they looked at fasted animals, autophagy was elevated. Every time the animals were fed, it dropped. Simple, clean, logical. Except it wasn't the whole story. Because as the research got more precise, as scientists started looking not just at whether animals were eating, but at what specifically they were eating, a far more nuanced picture started to emerge. It wasn't the absence of calories that triggered autophagy. It was the absence of specific molecular signals. And those signals, they come from specific amino acids. That single insight, that distinction between calories and signals, is the crack in the wall that lets all the light in. Because if you can control the signals, you don't need to control the calories. All right, let me introduce you to the molecule that is, right now today, probably standing between you and your body's ability to repair itself. Even if you eat well, even if you work out, even if you get your sleep and drink your water and do all the right things. It's called mTOR, mechanistic target of rapamycin. Don't worry about the name. Just understand what it does. mTOR is essentially a cellular foreman. When it's active, when mTOR is switched on, it tells your body, We're in growth mode. Build, accumulate, synthesize. Muscle gets built. Proteins get assembled. Everything is pointing toward expansion. When mTOR is switched off, the foreman goes home. And that's when the cleanup crew, autophagy, finally gets to come in and do its work. Here's the critical thing to understand. Growth mode and repair mode cannot run simultaneously. It's one or the other. Your body is constantly making that choice based on the signals it receives. And one amino acid, more than any other single molecule in your diet, is the primary trigger that tells mTOR to switch on. That amino acid is leucine. Leucine is what's known as a branched-chain amino acid, and it is absolutely everywhere in the standard Western diet. Chicken breast, beef, eggs, dairy, whey protein. All of these foods are loaded with leucine, and your body is exquisitely sensitive to it. A study published in Nature Metabolism showed that even small amounts of leucine, amounts well below what you'd find in a typical high-protein meal, are sufficient to fully activate mTOR signaling in the liver and muscle tissue. Think about what that means for a second. That protein shake you had this morning to support muscle recovery? It didn't just feed your muscles. It also, simultaneously, shut down the cellular cleanup crew that could have been repairing damage from the night before. Now, leucine doesn't work alone. Its BCAA cousins, isoleucine and valine, also nudge mTOR in the same direction, just with less force. And then there's another player, methionine. Methionine is a sulfur-containing amino acid found in high concentrations in animal muscle tissue, beef especially, but also eggs and poultry. Methionine activates a separate but equally potent blocker of autophagy. IGF-1. Insulin-like growth factor 1. Think of IGF-1 as a second lock on the same door. mTOR is lock 1. IGF-1 is lock 2. High methionine diets keep both locks engaged, and here's the part that most people, even most health-conscious people, have never considered. You don't have to stop eating protein to open those locks. You just have to eat different protein in different proportions. That's the blind spot. That's the metabolic loophole that nobody talks about. So let's get into the actual formula, because I know that's why you're here. And I don't want to bury it in jargon. Your body has internal sensors, molecular systems, with names like AMPK and GCN2 kinase that are constantly monitoring the composition of amino acids circulating in your blood. Not just how much protein you ate, what kind of protein, and in what proportions. Specifically, these sensors are tracking the ratio of essential amino acids, the ones your body can't make and must get from food, to non-essential amino acids, the ones your body can produce on its own. When that ratio tips heavily toward essential amino acids, which is what happens when you eat a steak or a chicken breast, the sensors read abundance. mTOR fires. Autophagy stops. But when that ratio shifts, when non-essential amino acids make up the larger portion of what's circulating, the sensors read something closer to scarcity. And scarcity is the language autophagy speaks. Research from the Salk Institute and corroborating work out of the USC Longevity Institute has helped clarify what serious longevity scientists have been converging on. When non-essential amino acids represent roughly 60 to 70% of your total amino acid intake, and when leucine is kept below 8% and methionine below 5%, your body can enter and sustain an autophagy state without any caloric restriction at all. You held that right. You're eating, you're satisfied, your body thinks it's fasting. Now here's the piece about methionine that most people completely miss, and it's important because it addresses the one thing everyone worries about when they hear restrict this amino acid. They think, won't I lose muscle? Fair concern, but here's what the research shows. Methionine and cysteine, another sulfur-based amino acid, share a metabolic pathway. Your body can convert one into the other. And when you restrict methionine while keeping cysteine intake adequate, you get the drop in IGF-1. You get the reduction in mTOR signaling. But you don't get the muscle wasting that pure methionine restriction causes. It's like finding the exact thread in a sweater that, when you pull it, unravels only the parts you want gone. The cell metabolism study that illustrated this most dramatically showed that mice on a low methionine, adequate cysteine diet showed autophagy markers equivalent to animals that had been fasted for 36 hours. 36 hours of fasting, without missing a meal. And then there's one more concept I want you to understand, because it's elegant. Researchers call it the dilution effect. Certain plant-based proteins, hemp seeds, spirulina, specific legume combinations have naturally low leucine concentrations and high proportions of non-essential amino acids. When these proteins make up the majority of what you're eating, your body's nutrient-sensing machinery, specifically a protein pathway called SESN2 that reports directly to mTOR, fails to register abundance. It registers scarcity. The gate opens, the cleanup begins, all while you're sitting down and having a real meal. Okay, theory is done. Let's talk about what this actually looks like on a Tuesday, because a protocol that only works in a lab isn't a protocol, it's a paper. I'm going to walk you through a practical 24-hour framework I call the Autophagy-First Protocol. And I want to be upfront. This isn't rigid. It's a structure you adapt to your life, not the other way around. The morning window. Extend what's already happening. Here's something most people don't realize. When you wake up, your body is already in a mild state of autophagy. You've been fasting for 7 to 9 hours. The cleanup crew has been quietly working through the night. Your first job in the morning is simple. Don't slam the door on them before they finish. That means no leucine-dense proteins first thing. No eggs, no whey shake, no Greek yogurt. Not yet. What can you have? Black coffee. Genuinely useful here, not just as a caffeine delivery system, but because caffeine independently activates AMPK, one of the primary pathways that upregulates autophagy. Multiple studies have confirmed this. Your morning coffee is, quite literally, pharmacologically relevant. Green tea, EGCG, the primary catechin in green tea, acts as a mild mTOR inhibitor. Again, not a supplement trick. Actual documented mechanism. Fruit, non-starchy vegetables, small amounts of hemp seeds or spirulina if you want some protein without blowing the amino acid ratio. You're not white-knuckling a fast. You're eating. You're just eating strategically. The midday meal, the bridge. This is your first substantial meal of the day, and it's where the amino acid ratio does its heaviest lifting. Build it around plant proteins with naturally favorable profiles, lentils and rice. Separately, neither is a complete protein. Combined, their amino acid profiles complement each other beautifully, and the resulting leucine concentration is significantly lower than an equivalent portion of chicken or beef. Hemp seeds, genuinely one of nature's underrated foods for this purpose. Complete protein, leucine content sitting at roughly 6% of total amino acids, right in the target zone. Pumpkin seeds, high in non-essential amino acids, notably glycine, which research suggests may actually support autophagy signaling rather than suppress it. Chickpeas with tahini, another favorable combination, and it actually tastes good, which matters more than people in the health space like to admit. Load this meal with olive oil and non-starchy vegetables. Fat doesn't activate mTOR. Vegetables don't trigger nutrient sensors. You will be full. Your cells will be cleaning. Both things can be true at the same time. The evening meal. The strategic reload. Now here's where it gets counterintuitive. This is the window where you deliberately reintroduce higher quality, higher leucine protein. And the timing isn't arbitrary, it's based on a simple logic. You've spent the day preserving and extending an autophagy-favorable state. Your cleanup crew has had hours to work. Now, you give the construction crew their shift. Muscle protein synthesis happens primarily overnight, during sleep. By loading your higher quality protein in the evening, you're sequencing your biology, repair during the day. Rebuild at night. The ideal evening protein for this protocol? Fish, particularly salmon, mackerel, or sardines, because the amino acid profile is excellent. The methionine content is meaningfully lower than beef, and the omega-3 content itself has independent anti-inflammatory effects. Pair it with complex carbohydrates, sweet potato, quinoa, legumes, to modulate the insulin response and reduce overnight cortisol. You're eating dinner like a normal human being, and your body is spending the night doing exactly what it's supposed to do. I want to be clear, the food protocol we just discussed is sufficient on its own, but if you want to push the signal deeper, 3 compounds have genuine mechanistic evidence behind them. Spermidine, found naturally in wheat germ and aged cheese and increasingly available as a supplement, has been shown to directly induce autophagy through a pathway that is independent of mTOR status, meaning it works even when mTOR isn't fully suppressed. Urolithin A, a metabolite produced when your gut bacteria process compounds from pomegranate and certain berries, specifically triggers mitophagy, the autophagy of damaged mitochondria. Now available in supplemental form for people whose gut microbiome doesn't efficiently produce it. Berberine, an AMPK activator with decades of research behind it, effectively mimics some of the cellular signaling effects of caloric restriction. None of these are magic. None of them replace the dietary foundation, but they compound the signal in the right direction. Let me be straight with you here. I'm not going to tell you that you'll feel like a different person by Thursday. What I will tell you is what the research actually shows, and roughly when, because you deserve real information, not a sales pitch. Days 1 through 7. The quiet shift. The first week is mostly happening beneath the surface. Your body is recalibrating. The nutrient-sensing pathways are adjusting to a new normal. Some people feel a mild passing fatigue as the adaptation occurs. Most people feel nothing unusual at all. But under the hood, autophagy flux is increasing. Damaged proteins are being flagged for recycling. Your mitochondria are beginning to undergo quality control, a specific subset of autophagy called metophagy. The cleanup crew is clocking in. Days 8 through 21. The shift you can feel. This is typically where people start noticing something. Not dramatically, not overnight. But the afternoon energy crash that you assumed was just part of being an adult? It starts to ease. Mitochondrial efficiency improves, and the difference shows up as steadier energy throughout the day. Skin texture and elasticity often begin to improve, because skin cells are among the most autophagy-active cells in your body, and accelerated cellular turnover has visible consequences. Inflammation markers, specifically IL-6 and CRP in blood panels, typically begin to move in the right direction. Days 22 through 30. The systemic reset. By the end of the first month, the research on autophagy-enhancing dietary patterns consistently shows measurable improvements across multiple systems. Insulin sensitivity improves because autophagy clears the dysfunctional protein aggregates that physically impair insulin receptor function. Cognitive sharpness increases. Neurons are among the most autophagy-dependent cells in the human body. When the cleanup happens, they perform better. Sleep quality often improves, tied directly to mitochondrial health and reduced neuroinflammation. And here's how to actually verify. It's working. You don't have to take this on faith. You can measure it. The GKI, the glucose-ketone index, remains one of the most reliable indirect markers of autophagy-favorable metabolic states. A GKI reading below 3.0 correlates strongly with active autophagy. A continuous glucose monitor paired with a finger-prick ketone meter gives you this data in real time, and both have become genuinely accessible and affordable. IGF-1 blood testing, a standard lab panel available through most direct-to-consumer testing services, directly measures one of the 2 primary autophagy-blocking hormones. You want to see it sitting in the lower normal range. BCLIN1 and LC32, specific autophagy protein markers, are now becoming available through specialized functional medicine labs. These are as close to a direct window into autophagy flux as currently exists outside of a research setting. Track these. Watch them move over 30 days. That's your data, not a guess, not a feeling. Actual feedback from your own biology. Now let's land the plane. Your body has a sophisticated, Nobel Prize-validated repair system built into every cell you own. For years, honestly, for decades, we assumed the only way to activate it was to stop eating entirely. We were working with an incomplete picture. The actual switch isn't calories, it's signals. And the most powerful signals come from the ratio and composition of amino acids your body detects. High leucine, high methionine, high essential to non-essential ratio. mTOR activates. Growth mode. Autophagy. Door slams shut. Low leucine, restricted methionine, non-essential amino acids dominant. mTOR quiets. AMPK takes over. The door opens. The practical version of this isn't complicated. In the morning, extend your natural autophagy window with coffee, green tea, and low leucine foods. At midday, build your meal around plant-based proteins, lentils, hemp, chickpeas, that naturally hit the favorable ratio. In the evening, reload with higher quality animal protein, fish is ideal, to drive overnight muscle synthesis. Layer in Spermidine, Berberine, or urolithin A if you want to deepen the signal. And track your GKI and IGF-1 so you're working with data, not guesswork. That's the whole thing. The longevity benefits of chronic fasting, without the cortisol, without the muscle loss, without canceling dinner plans because you're on day 2 of a water fast. Just your body, running the protocol it was always designed to run. Now, there's one more layer to this that most people completely overlook. Your circadian rhythm directly modulates how efficient your autophagy actually is. The same meal eaten at different times of day produces dramatically different cellular outcomes. Same food, different clock, different biology. That's exactly what we're getting into next time. Make sure you don't miss it.